Hey everyone, thanks for the invitation to talk. I'm going to talk to you about this collaboration we've been leading here in Aberdeen, taking up a large part of my work for the last few years. So just to outline what I'm going to talk about today, introduce you to Parkinson's disease and prognosis in Parkinson's. Talk a bit about methodological issues in previous studies of prognosis. Talk a bit about the PINE study, local study in Aberdeen. Then go on to talk about the methods and the results of the PIC collaboration, and some reflections on the process and challenges of leading this collaboration. So Parkinson's disease is the second commonest neurodegenerative disorder. It affects a large number of people in the UK and indeed worldwide, and it's a disease that's predominantly affects the elderly, and as the population's age, it's becoming more common. It's caused by a progressive loss of brain cells, producing a particular chemical called dopamine, though other parts of the brain are also affected. It's a progressive disorder, so gradual deterioration over time. This often leads to disability, difficulty with even basic activities of daily living, and various complications, most notably and probably most feared is dementia. At the moment, we've got no treatments that slow down disease progression, no treatments which slow down the loss of brain cells. But there are good symptomatic treatments, so we can replace the dopamine, this chemical that is deficient, with tablets, and that can lead to quite a marked improvement of the symptoms of Parkinson's disease. But we can't slow down the ultimate progression of the disease. Just going to add a note at the bottom here about Parkinson's disease versus Parkinsonism. Some of the results I'm going to show you refer to Parkinsonism, which is an umbrella term for a group of conditions, including Parkinson's disease being the most common of these, and other conditions which share a lot of features with Parkinson's disease, but are less common and generally more aggressive than Parkinson's disease. So I'm just going to show you a couple of videos to highlight the point here that there's huge variability in prognosis between different individuals with Parkinson's. So these are two people with the diagnosis of Parkinson's disease. Both are 5 years from the time of diagnosis, so they're the same time at least from diagnosis. The chap here on the left, seeing him in the research clinic. You can see him walking here. He's got a tremor. Tremor is a very common feature of Parkinson's disease, and but he can walk well and he's independent in functioning well in daily life. This chap, on the other hand, is in a nursing home, and here Carl is examining him. You can see he's got a catheter in. He's unable to stand up from the chair, and he's got a severe dementia. And I should point out there's similar ages, these two gentlemen at the time of diagnosis. The one on the left is a few years younger. So this presents a problem for us as clinicians and a challenge for us as researchers. Can we understand better the variability in prognosis? Can we make better predictions? And can we even alter the prognosis so that fewer people are like this chap on the right and more people like the chap on the left? So why should we study prognosis? I should perhaps say prognosis, just to define it more explicitly, is how people fare over time with a particular disease or condition. And if we know better, if we have more detailed or more accurate information about prognosis, this means we can tell people who are affected by the disease, the individuals with the disease, their relatives and their carers, how they might be affected over time. And that's important for lots of reasons, not least being able to plan for the future. We may be able to identify prognostic factors, factors which influence prognosis that we can modify and thereby improve prognosis. We can use information about prognosis to guide treatment decisions. It may be that somebody with more aggressive disease, you're going to give more aggressive treatment that might come along with more side effects and more toxicity. And there's benefits in research, particularly in the field of clinical trials. You can use information about prognosis, particularly if it's encapsulated in the form of a prognostic model, to select patients for inclusion, to stratify, randomize. So selecting patients for inclusion, you could select a group of people with more aggressive disease to study a more powerful, more toxic treatment. You could stratify randomization, ensure that that the groups, so if you randomize groups into a randomized trial, you'd expect that they would be the same in their characteristics, but you can get differences by chance. And if you stratify randomization, you can ensure that both groups, or the groups that are in the trial, are similar in respect to prognosis at the start of the trial. You can also use prognostic models to adjust analysis for confounding in a trial, and that can lead to great efficiencies in a trial. So there's an example where in a head injury trial, using a prognostic model to adjust the analyses was predicted to reduce the sample size by about 25%. And if you think that randomized controlled trials often cost many millions to carry out, that can lead to huge cost savings. Also important for service evaluation, if you want to make a comparison between different hospitals, or say between different surgeons in terms of their outcomes, you want to adjust that for the prognosis of the cases that are seen in the hospital or by the particular surgeon. So a more experienced surgeon may take on riskier cases which have got worse outcomes. And if you want to make any meaningful comparison, you would have to adjust for prognosis. And really, these benefits need a personalized estimate of prognosis. It's averages are all very well, but I think this is encapsulated nicely by this quotation from Stephen Jay Gould, who was an evolutionary biologist, and said that the median's not the message. He was diagnosed with a very rare type of cancer, and he was told that the median survival was a year or 2 years or something. And then 30 years later, when he had survived being cured from the cancer, he was pointing out that the average wasn't particularly applicable to him. And this can be best done with prognostic models. And one of the, as Simon mentioned, one of the things I've been doing is trying to develop better prognostic models in Parkinson's. So I want to come on to talk a bit about the previous literature that is out there about prognosis in Parkinson's. And you try to illustrate some of the methodological issues that have led on to some of the subsequent work I want to tell you about. This is a figure that's taken from a systematic review I did as part of my PhD, looking at mortality or survival in Parkinson's disease. These coloured lines all represent different studies, cohorts of people with Parkinson's disease that reported survival. And to me, the striking issue here is the heterogeneity between the studies. So if you were wanting to work out what the average survival in Parkinson's disease is after 10 years, you could, if you like, say, quoting this yellow study at the top, that was 90% of people survived 10 years of Parkinson's. There's another study down the bottom here, which is about 25% of people surviving Parkinson's, and a huge range of values in between. Now, these cannot all be the true value, as it were. You could take a simple average of these things, but that might lose some important information about why there are such differences between the studies. So one of the things I tried to do is to work out why there are such dramatic differences between estimates of mortality in different studies of Parkinson's. So this graph has got some of the same information as the previous graph and some additional information. So again, we've got studies here that reported survival in Parkinson's disease. I've removed all the studies that recruited patients at varying times after diagnosis, and just kept here the studies that have recruited patients from diagnosis or about diagnosis. So these are early disease cohorts, recruiting people after the diagnosis and following them up, which I think is the only sensible way to study prognosis in a condition, unless there are specific research questions that you want to ask in particular groups of people. I've also added in some single study estimates. I left them out of the previous slide because there was so much information there. So the lines connect repeated estimates of survival within single studies, and the single dots are studies which reported mortality or survival at a single time point. But even if you just look at these studies that follow patients up from the time of diagnosis, again, there's still dramatic variability. We've got a study of 90% up at the top there, and just under 40% with 10-year survival. So again, this reduces some of the heterogeneity, but it doesn't really explain the large part of the heterogeneity. But the other thing I looked at was where these cohorts, or how these cohorts were derived. And I've split them here into blue being hospital clinic-based studies and red being population-based studies. So the studies in blue, which have been many of the studies in Parkinson's, have recruited patients from a specialist clinic where they were seeing people with Parkinson's, which is in many ways an obvious place to recruit people, because you've got lots of people with Parkinson's in one place. But these are largely done in very large tertiary referral academic university clinics, and the types of people that go to these clinics may be quite different from the general population of people with Parkinson's. And the red studies are those that made an effort at least to try and recruit everybody in the population into the study, which gives a much more representative sample of patients. And you can see, and I was surprised how clear the demarcation was between these two groups of studies. You can see that there's an almost perfect demarcation between these studies, apart from at this point here, where there's two studies that are actually the same value in the two studies, so I've separated the points slightly. So clearly, a large part of the variability between the studies depends on whether the cohorts recruited a general population with Parkinson's or recruited those coming to a special clinic. And one of the big reasons in Parkinson's why this is such a dramatic effect is that neurologists, particularly highly specialist neurologists, tend to see the younger, the fitter people with often more complex disease, and the older and frailer will often be seen by either general neurologists or geriatricians. So there's big referral biases in how people get to these specialist clinics. So this tells us that methodology is important when you're studying prognosis. And I've mentioned some of these selection biases. I think different disease durations and entry to study is an important one. Studying selected patient groups from specific hospital clinics, I think, is probably the biggest factor. Another issue relevant to Parkinson's is diagnostic accuracy or diagnostic inaccuracy. I mentioned at the start that there are various different conditions that can look like Parkinson's, so it can be difficult to distinguish these, even by experts following people up lifelong. It can't be done perfectly, and studies that use routine data to identify Parkinson's can have a very high diagnostic inaccuracy problem. So how do you get around this? Well, I hope I've convinced you that representative cohorts that study everybody with Parkinson's from diagnosis is probably the best way to do this, together with careful reassessment of diagnosis over time. And those of us in clinical practice must bear in mind that the data we use to inform prognosis in our patients must be generalizable to our patients. And that's best done with population-based studies. So Carl Council set up the PINE study back in 2002 which is an incident cohort of Parkinsonism in Aberdeen, collecting all new cases of Parkinsonism, so Parkinson's disease and related conditions that can look like Parkinson's, over a 4.5 year period in total, using various methods to try and identify everybody in the population with Parkinsonism. Diagnosis was done with the appropriate diagnostic criteria, and the diagnosis will be evaluated at least every year prospectively, and post-mortems were also done to try and confirm the diagnosis where participants agreed to that. Patients were assessed every year and at home if unable to attend clinic. So everything was done to try and maximise the quality of the data collection in this study. Remarkably, it was a 94% consent rate to follow up. That's really unusual. And very few losses to follow up. When it comes to vital status, no losses to follow up in the Parkinson's group, at least, and very high data completeness for other outcomes as well, though not 100%. And I'm just going to show you some of the results that have come out from the PINT study over the years. This is another piece of work that I did as part of my PhD, looking at dependency or functional dependency in Parkinson's. So by dependency, I mean needing help with the basic activities of daily living. So things like washing and dressing and toileting and mobility. So if you're independent, you can do all these things yourself. If you're dependent, you're going to need help from somebody, whether it's a spouse or professional carers, to help with these things. And what we found, which hadn't really been understood before, how it really been an area that was neglected in Parkinson's research up until this time. And this problem is really common. And even at diagnosis, about a fifth of people are dependent, so need help with activities of daily living. And by 10 years, a clear majority have either died or are dependent on others for these basic activities of daily living. And those who are independent at baseline, the median time to becoming dependent was just over 5 years. So this is clearly a big problem. It's an important thing to study, and maybe a useful outcome for clinical trials. It's got advantages. It's easy to measure. It's common, and it's really very relevant to people with Parkinson's, unlike some of the more commonly used outcome measures in clinical trials in Parkinson's disease. Another study that Isabel Sleeman did, when she was a junior doctor here, she looked at fractures in the PINT study. So this is a Kaplan-Meier plot of fracture-free survival in people with Parkinsonism and in controls, and that includes Parkinson's disease and these related conditions. And it's clear that fractures are much more common in people with Parkinson's than in controls, that I haven't shown the data here, but the incidence of fractures in Parkinson's disease is very similar to these other conditions. And this is a huge problem in Parkinson's, particularly hip fractures. Often have a high mortality rate and a high morbidity rate. So these are important. These are really big problems for people with Parkinson's, and it's something we need to be doing clinically, is assessing people for fracture risk, and people who are falling in particular, trying to ensure they don't have osteoporosis, or that they therapy input to try and reduce the risks of further falls. So that's a clinically important finding. Another piece of work that we have done is looked at delirium in Parkinson's. So delirium is a confusional state that people develop, often triggered by infections and other health problems. that makes people confused and is a serious problem, and is known to be associated with in general in people in hospital. It's a marker of reduced brain reserve and is associated with clear increased mortality. So it's a sign that somebody's vulnerable and may not do so well. This hasn't really been studied in any detail in Parkinson's disease. So this was the first study to look at the incidence of delirium in people with Parkinsonism from diagnosis. This is just comparing people with Parkinson's and those with atypical Parkinson's. Developing delirium is common in both these things, much more common in the other forms of Parkinsonism. And once people have delirium of Parkinson's, the graph on the right, there is a high mortality rate. So the average survival after developing delirium is about 2 years. This is an important prognostic marker that we should be paying more attention to in Parkinson's disease. Another interesting thing that I looked at when I was doing my PhD was mortality and life expectancy in Parkinson's. So it's a bit difficult to study life expectancy for various reasons. But looking at the median survival here in people with Parkinson's in different age groups under 65 to 74, 75 to 79, 80 plus, you can see that the median survival drops dramatically as you go up these age groups. So those under 65 have better survival than the older groups. But if you compare that with actual life expectancy, and these are derived from life statistical data from Aberdeen and Aberdeenshire, you can see that proportionally, actually, even though the survival is better at the younger age groups, the proportional amount of life lost from Parkinson's is substantially higher in the younger group of people. So that's a message that hasn't really got out to the Parkinson's community. For there's obviously difficulties in sharing this information with people with Parkinson's. So the younger people with Parkinson's have more to lose, unfortunately. So these are some of the things that we did in just some examples of work done in the PINE study. The PINE study is relatively small. There's about 200 people with Parkinson's. That has various limitations. And we were aware of some other studies out there that were similar to the PINE study, that were incidence cohorts and had followed people from that incidence study up long term to study prognosis. So we did a systematic review. The aims were we tried to identify all such prospective incidence cohorts. So studies that start with an incidence study and then try and follow up as many of these people as possible. That gets everybody or tries to get everybody with the diagnosis in the population, and to pool the data from these studies and to study long-term prognosis. And this led to the Parkinson's Incidence Cohorts Collaboration, or PIC. And we carried out a systematic review of incidence studies in Parkinson's disease and identified which of these studies that we included had long-term follow-up of the incident population. And there are 6 such studies. They're the only studies we've been able to find in the world that have been designed like this. They're all in Western Europe, UK and Scandinavia. 2 studies in Cambridge, a study in Newcastle, a study in Umeå. I can't pronounce that word properly, I'm afraid. So apologies to any Swedes who might be listening. In the north of Sweden, and Park West, which is based in Stavanger in Western Norway, as well as the PINE study in Aberdeen. And how did we do this? We got funding from Parkinson's UK, which is a support organisation for people with Parkinson's that does quite a lot of research funding. So we got funding from Parkinson's UK for an initial investigator meeting in London. So 2 investigators from each of these studies met together to discuss collaboration. We worked out what data there was in the cohorts that we could pool together. I'll speak a little bit more about the data pooling, and develop various agreements to govern the collaboration. So there's no overall collaboration agreement, data management plan, and a data request form that individual researchers in the studies involved could submit, so that any requests for access to data and were evaluated and approved by all the centres involved before the data would be used. So a big part of the work involved in this was data pooling and harmonisation. And apologies for a slide with lots of text in it, but it reflects how I feel when I look back on this process of data harmonisation. It was pretty heavy going, and it's not the most exciting thing in the world to be ploughing through data. We received data sets from each cohort, different formats, different formats in terms of file types, different formats in terms of the layout of the data. Some were long, some were wide. Lots of differences in variable names between the different studies, which needed to be standardized. There was issues about consisting of the coding of the variables. So how was sex coded, what was 0, what was 1, what was 1, what was 2, etc. Genetic variables, there were some differences in how these were coded between studies. So that had to be, these things had to be sorted out. We had to work out how each study defined the different variables that were used, and there were differences, how important these were. So one example is the definition of dementia. There are different clinical criteria that are used to define dementia, which were different in different studies. Probably not a hugely important difference, because we're all the investigators in all the studies are trying to identify the same thing, but these may lead to heterogeneity between the studies. There's different versions of scales in one particular scale in particular measures impairment in Parkinson's. There's a published conversion factor, so you can convert from one version, the old version, to the new version of the scale. And we didn't have a common measure of functional dependency between the studies, and we believe that this was an important outcome to be studying in PIC. So we developed a new measure of functional dependency and validated that as part of this process. I'll tell you a little bit more about that in the next slide. The various data sets were then appended into a single pool database with data at baseline, and then follow-up data at various time points up to 10 years from diagnosis. And this 1,100 participants in total, including some controls, and about 2,400 variables, and that's mainly because lots of these variables were repeated at each year of follow-up. So it was a fairly cumbersome database, and a lot of variables having to be sorted out. And what I think was the data management team, or Dash, were very helpful and did a lot of this work. A lot of the decisions needed clinical input, because to know what the variables were and so something I certainly hadn't realised when I started this is how much input I had to have into this process. Developed a data dictionary so that we could be explicit about what the various variables in the database were, and as I mentioned already, a lot of this work was done by the data management were Arthur and Adi. So one of the things we did as part of this process was to validate a measure of dependency, and this was because we didn't have a common measure across the cohorts, across most of the cohorts. So we used information from a different scale that combined some measures of activities of daily living together with lots of other things, and these items were relevant here were feeding, dressing, hygiene, so washing, walking, and getting out of a chair. And we developed a threshold based on the sum of the scores from these different items, and tested validity. So we looked at concurrent validity. Did it correlate with other measures of dependency that were in some of the studies? Convergent validity, did it correlate with similar measures? So at the top here, the writing's very small, but we've got a quality of life measure, cognitive measure, an impairment measure, and in each of these, there are worse scores in the dependent than the independent group. And we also looked at predictive validity. Did dependency status influence mortality? And we've done that at different time points. Here's data from baseline in year 5, and there was clear difference in mortality between these two groups. So clearly there this gives some evidence for the validity of this measure. So that's a bit of the process we went through to pool data. Here we have an overview of the studies that were included in this collaboration. And some of these things I think are interesting in terms of thinking about some of the differences between the study. They're not all homogeneous, and I think there's some differences in the quality between the studies. So different time frames, some more recent than others. I don't think that's a particularly important issue. Consent to follow-up rate is important, and we're trying to say these studies are identifying as many people as possible with the diagnosis of Parkinson's in the population. You want most of the people to consent to follow up, and 2 studies were much higher than the others. So that's our study here in Aberdeen and the Swedish study. One study was a bit lower, 48%. I think it's a bit questionable how representative that study is of the population of Parkinson's in general. But they were reporting at least to be an incidence cohort. So we've included them in the collaboration. And different sample sizes, all relatively small. It's a very labour-intensive process to go through to recruit and follow up an incidence cohort. And 883 true incidence patients, and there's some other people as well that 2 of the studies had some inception but not incidence patients. So patients that were at the time of diagnosis recruited but not as part of the incidence study, so not as part of a formal. Couldn't be sure that it was these were everybody in the particular population or time frame. So it's these have been included in some of the analysis, but not in others. Follow-up varies over time in terms of the data we have in the collaboration, with the median and maximum follow-up. Okay, so some of the studies that have come out of the PIC collaboration, this work was led by collaborators in Cambridge, looking at constipation. So constipation is a very common feature in Parkinson's. It's one of these interesting things that can occur many years before somebody gets Parkinson's, which is telling us something interesting about the disease process and pathophysiology. What is the significance of Parkinson's? Is it just an inconvenience for the people who experience it, or is it something? Does it tell us something more? Well, we've looked at this in PIC, and constipation severity is associated with a high risk of developing dementia. Hazard ratio 1.45. And the Kaplan-Meier plot shows you the difference between the groups. So major constipation certainly has the highest risk of dementia versus minor and no constipation. What does this tell us? It probably tells us something about the role of the gut and the gut-brain axis in Parkinson's disease. But I think a lot more work needs to be done to tell us more about that. We've also looked at some genetic data. So all the studies had genetic data, and here we've looked at 2 particular genetic markers which are predictive of cognitive decline. So this is in 1,000 PD patients, followed for 7.2 years on average. ApoE epsilon 4 variant and mutations in the GBA gene, I won't go into the details about these things, are associated with higher hazards of dementia, and probably an interaction between these 2. So having the ApoE variant has almost a 3.5 times higher risk of dementia. Now that's known, and that variant is known to be a risk factor for Alzheimer's dementia. So it could be telling us something about co-occurrence of Alzheimer's disease and Parkinson's disease causing dementia, and GBA mutations, which are associated with more aggressive Parkinson's on average, has a less strong effect, but has does seem to have a statistically significant effect on developing dementia in Parkinson's. And this is a piece of work that I'm working on and hoping to publish very shortly. This is looking at describing outcomes in terms of what I think are probably the most important outcomes in Parkinson's disease over time. We've got the top right-hand corner, probability of this is to do with disease staging, but it's essentially it's time until somebody starts to have balance impairment in Parkinson's and start to fall. We've got remaining independent to the top right, which I talked about already, probability of remaining without dementia, probability of remaining alive in Parkinson's. So I think these things encompass 4 vital outcomes in Parkinson's. So we've studied these in the PIC cohort. We've also looked at predictors of these outcomes, which I don't have data. I'm not going to show you data for. But there's some interesting things looking at this. I think, given the graphs I showed you of mortality previously, these studies are much more consistent in their results than the studies I showed you before. There is notably some heterogeneity, but they're fairly homogeneous, these studies. Some of the dementia is relatively homogeneous, but there's some more variability. There's various reasons for that. But some of the other outcomes, there's still quite a lot of heterogeneity, and I'm not quite sure why that is. I think part of the difficulty with the dependency measure is heterogeneity and how the different items that were used to develop that are scored between individual centres. And the scale that we've used to define balance impairment is a confusing scale to use. So some people with Parkinson's could fall between 2 categories on the scale, so there's some subjectivity. But that is a problem, I think, this heterogeneity, and it's something that would be interesting to look into further. But the more concrete, hard outcomes, we've got much more consistent data between the studies. In fact, we've developed, I think, useful data about in terms of prediction. So this slide shows the probability of remaining alive in Parkinson's by age and sex. One of the interesting things about Parkinson's is that there's a sex difference in survival. So men do worse than women. It's also, in terms of the incidence of Parkinson's, it's about 50% more common in men than women. We don't really know why this is, but there are clear sex differences in Parkinson's. But I think this is relatively useful information that we can use for people with Parkinson's who want to know more information about how about the future in Parkinson's. So there's more sophisticated things we're working on, but this is at a very simple level, combines age and sex, and we can use it to discuss probabilities of survival, say at 5 years and at 10 years from people with Parkinson's disease. And using this data, I've developed infographics to try and make that easier to share with people with Parkinson's. And we're working with Parkinson's UK, which is the main charity supporting people with Parkinson's, to try and work how we can share this with people with Parkinson's in a sensitive way without causing upset, and while giving people some hope despite what may be difficult news or disappointing information for them. We're also working prognostic modelling in PIC. David McLernan has done a lot of this work. I don't have time, I don't think, to go into this in detail, but using formal individual patient data, meta-analysis to do the prognostic modelling, and there is some heterogeneity in how how the prognostic model performs between the studies. And we have a PhD student, Jan, who's working on dynamic prediction modeling, which is proving to be challenging for various methodological reasons. But the idea here is that you use the information that develops over time to refine predictions of future outcomes. So you've got this is related to dementia. So over time, you make predictions at baseline, and then subsequent years, as things change, such as cognitive function and severity of the Parkinson's, you can refine the estimates of prognosis. So that a lot of prognostic models just use information at time of diagnosis, where this is a dynamic process that updates the information over time. So, but one of the practical issues here is that the different studies have different timings of follow-up. So Pine and some of the other studies saw patients every year. Some of the other patients, some of the other studies followed up patients less frequently, and there's this heterogeneity between studies in terms of when the data is available is proving challenging when we're doing the prediction modeling. So that's something that's probably going to need further data to achieve effectively. So to go on to talk about the practical challenges. So thinking about some reflections on this process and this collaboration. It's been a slow process. Part of that's my fault. Other things on my plate, and some procrastination along the way, I must confess. And the data harmonization process took much longer than planned. It was more work than I had expected. So perhaps I went into this somewhat naively. It required a lot more involvement of somebody with clinical knowledge. If I was going through this process again, I would definitely want funding for more research assistants or people with, ideally somebody with expertise in data management and expertise in Parkinson's to do this process. And this had somewhat limited funding, and I think again, if I was doing this again, I would try and get more funding and more realistic estimates of what funding would be needed to do this more efficiently. There have been some challenges encouraging interest from the different members of the collaboration. All the different academics involved have got various priorities, and the PIC hasn't always been the top priority for people. It's been an important part of what we've done to try and encourage interest along the way. So we've had various meetings, usually to coincide with international conferences where most of us have been attending, and I think that's been a helpful thing. And going out for a meal together, practically that does help. And it's nice to actually sit in a room and chat with people as opposed to just discussing people over Teams, which is how we meet more commonly. Arranging any meetings is always a challenge. You've got different people with busy schedules and different time zones as well, and that has always been challenging. And the way we've worked that is trying to get at least one person from each cohort available, even if not everybody is available, and that takes a bit of time to plan. Academic challenges. I think one of the big issues here is there's inevitably competing interests between what individual studies want to do themselves and publish, and what's in the interest of the collaborative group as a whole. And one of the studies in particular has given us pretty limited access to their data in terms of follow-up duration compared to what they have available, whereas other studies have shared all the data that they've got available. So some people are more are keen to keep some of the data to themselves to do the things they want to do, which is fair enough, but doesn't always work best for the collaboration. Authorship, we've not had any major issues about authorship. The way we've dealt with that is the authorship has to be stated on the approval form for planning each study. So at the stage of requesting the data, the authorship has to be stated. We'll talk a bit about data inconsistency between the studies. Differences in timing has been an issue when we've been trying to do the dynamic prediction modelling. Differences in the variables that are available. So some studies simply don't have data. So the constipation study I mentioned, there was only 2 of the studies that were actually included in that. Most of the outputs we've had from this have included all the studies. And I think perhaps a bigger factor is when it comes to predictive modelling, various important prognostic factors that we would like to study have not been available in all the studies. So that has been another limitation in what we've been able to do. One issue I think is relevant here is different levels of methodological rigor between the different people involved. So when the way we've approached this in Aberdeen is as a formal IPD meta-analysis and using methodology that explicitly accounts for the heterogeneity between studies when we've been doing the analyses. When some of the other outputs have just lumped together all the participants as if they belong to a single cohort, which statistically is not the best way to do things. And there's clear methodological literature about this. And that's an interesting challenge about how we deal with that. And the way we have dealt with that is whoever's leading a particular project takes the lead in these things. And I guess that means that I'm a co-author in some studies that haven't dealt with things as rigorously as I would have liked to do that. But I think that's a price to pay for encouraging the collaboration, which has certainly had various benefits, important benefits, I think. And another issue which will come up against recently is that I've done a paper and I've sent it around the various collaborators and suggesting that one study team have suggested that we add data from their study which wasn't previously shared, which is a little bit frustrating now that the analyses have been done and the work has been written up. So anyway, hopefully we'll work through that, and my plan is to submit the paper as is and work on the analyses in the meantime with the additional data if we can do that. So these are some of the challenges involved with doing this, but I think there's been important opportunity. I think we have developed genuinely useful collaborations, and this collaboration hasn't just done for the sake of collaboration by any means. It's been useful, and we've done useful research and important publications that have come out of this. Clearly, collaborating together brings greater power for statistical analysis, which I think is important. We would also be able to pool expertise. So we've particularly got epidemiology expertise in Aberdeen. Our Stavanger colleagues have particular expertise in genetics, which has been useful. It has led to us doing more genotyping of the Aberdeen DNA samples that we had in the Stavanger lab, which has been useful for some of the publications. I think collaborating with these other centres has undoubtedly led to higher impact publications and just doing things on our own in a single study. And I think I'm hoping that what we're doing is promoting higher quality epidemiology in the field of Parkinson's in general by trying to do things with high quality cohorts, which is obviously a challenging thing to do and resource intensive way to do research, but I believe it's important, and I hope that we're able to promote that. So I'll just finish with acknowledgements. Various study participants and staff running the studies, data management team who have been very helpful in this process, collaborators in Aberdeen and these other centres, and the funders, of course, who have funded this research. So thanks very much. Happy to take any questions.